All right, so today we're talking about protein function. We talked about structure last time, and so today we'll be looking at function. And I had a clicker at one point, but can't find it. Oh well. All right, so let's talk about some naming. I've already mentioned this before, that when you see the word enzyme, there it is. An enzyme is just a protein that's carrying out a biological reaction. Okay, so an enzyme is just a protein carrying out a reaction in a biological system. Now for us as chemists, if you were taking like general chem, a chemical enzyme, a chemical catalyst is different from a biological catalyst, right? So enzymes are speed up biological reactions, right? But when we talk about it as chemists, that's a little bit different. So a couple of vocab words we need to talk about. This is the protein, right? Enzyme's just the protein. And the active site is where the reaction occurs. Okay, you probably know this from your biology class, right? The active site is where the molecule comes in, and that molecule is called the substrate. Okay, so the reacting molecule comes into the active site, and then that creates what's called a conformational change, right? So the shape of the protein actually changes there. That causes a conformational change. And the R groups at the active site obviously would be significant, right? Because the R groups need to be compatible with that substrate. And so the substrate fits into the active site, creates a conformational shift that either breaks a bond, forms a bond. We'll look at all the different types of biological reactions today. And then once the reaction is complete, then the products are released. Okay, so here's a nicer picture, right? Just showing. This would be a quaternary structure, right? Because it's got 1, 2, 3, 4 subgroups here. And so there's the active site, right? And again, hydrogen bonding is what's holding this substrate in place. Now, are all active sites going to be maintained by hydrogen bonds? No, but they are going to be maintained by R group interactions, right? So what if you had a mutation that affected the active site? That could be a bad thing, right? It could also be a good thing. Because now what if your active site contains an amino acid that's even more compatible? Maybe that'd be good. What if your active site now contains an amino acid that's supposed to be nonpolar and now it's polar, right? That could be really bad. So again, we're looking at the effective mutations. You can't make one lump sum and say they're all good or they're all bad, right? You have to look at where it's affecting the protein, right? If it's affecting somewhere way down here that's nowhere near the active site versus up here at the active site, that's a different discussion as well. So this is a figure from your textbook. You've probably seen it in other biology books and that sort of thing as well. The enzyme substrate complex, right? So here's our substrate, here's our enzyme, right? That's what the active site is. Now it's not an exact lock and key, okay? I don't want you to think about it like a lock and a key, because that's not really what it looks like. It's a flexible active site. So in other words, it adjusts. So the substrate comes in, it makes what's called the enzyme substrate complex that catalyzes the bond breakage or bond formation or whatever's going on, right? And then you've got your product and it's released, right? So it is flexible. And again, when the substrate comes in, it creates a conformational shift. So it actually changes the shape there. That's what we mean by flexible, right? So it looks like a triangle, now it's kind of got that different shape. So don't think about it like a lock and a key.
Flexible, can anything go in there?
Well, that's what we're going to talk about in just a second. Yeah. So enzyme specificity. Enzymes recognized by similar substrates, types of bonds, and this is called an inhibitor. So when you've got a molecule that is similar to the actual substrate, it can also go into the enzyme as well. And so a molecule that is a structural analog, so it looks structurally similar to the intended substrate, those are called enzyme inhibitors. So they would compete. So if you've got an enzyme and a substrate that's supposed to be there, and an enzyme and a substrate that's not supposed to be there, they would be competing for that active site. And there are two types of competitive inhibitors we're going to talk about, reversible and irreversible. So reversible competitive inhibitors, see there's the enzyme, there's its active site. Here's the normal substrate, and here's something with a similar shape. right? We call that a competitive inhibitor. When the normal substrate enters the active site, reaction goes on. Da, 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 da. When the competitor enters, no reaction, it just falls out. That's reversible, meaning it'll come out, right? So if both of these are in the cell, this one might pop in, and it says, oh, nothing's going to happen, and it just comes out, right? Obviously, it might slow down the activity, though, if this thing's in the way. You got to wait for it to come out. Right? So it could slow down the overall rate of the reaction if these are in there junking it up, and you got to wait for those to come out. But it can be removed, right? That's where we get reversible. Now, irreversible, obviously, that's a problem, right? Because now they're structural analogs, but the competitor binds irreversibly, meaning if the competitor comes in, it stays in, right? It doesn't come out. And now this enzyme is completely shot, right? If the competitor comes into the active site and doesn't leave, now you've got a problem, right? Because now this entire enzyme's out of function, right? Because its active site's jumped up with this competitor that's not leaving. And so a good example is carbon monoxide, right? It's structurally very similar to oxygen gas. There's O2's little structure, there's carbon monoxide, right? They're structurally very, very similar. Carbon monoxide binds about 200 times better to hemoglobin than oxygen gas, right? Oxygen gas is carried through our blood by hemoglobin at its active site. And so if carbon monoxide is in the blood, right, it's going to bind to hemoglobin, and it's about 200 times better. That's bad, right? This is why you have a carbon monoxide detector in your house, because it's odorless, colorless gas. And it binds irreversibly, right? So this is looking at the heme group from the side. There's O2, and there's an iron 3 in the middle, iron 2 in the middle, right? But it's just stuck on there. It's not coming off, right? So it's there. It's a competitive inhibitor that is irreversible. So do we see the difference between reversible and irreversible competitors? They have to be structurally similar. If it comes out, we call it reversible. If it doesn't come out, we call it irreversible. Does that make sense? Okay, so like we just said, 3D shape determines how functional it is. And so when 3D shape gets disrupted, obviously the function is going to be disrupted as well. And so there are two types of things that we talk about that can disrupt 3D shape. We classify them as hydrolysis and denaturation. I combine them into the same slide. You could just download it from the website, so you don't have to write all this down. Right? When you hydrolyze a protein, it's not going to be functional anymore, right? Because you're going to break it down. it's gone. It's no good, right? So you can hydrolyze a protein by pH, by using other enzymes, by temperature. But these are all things that you can chop up a protein with. Denaturing is when you don't actually chop it up, but you break apart the 3D shape. What's holding that 3D shape? The IMFs, right? Intermolecular forces between the R groups give the protein its tertiary structure, its quaternary structure. And so if you mess that up, right, you're not actually chopping it up, but you are disturbing those IMFs, which would cause it to fall apart, right? So temperature, metals, detergents, solvents. Mechanical stress, that's like beating an egg, right? That's what mechanical stress means. And so this is a figure from your protein, a figure from your textbook. There's your active protein, heat, acid, base, heavy metals, salt, agitation, whatever, right? When you take your raw egg and you heat it, you're not breaking the bonds, you're just denaturing it, right? You're destroying that three-dimensional shape. And so now it doesn't have those tertiary structures anymore. It's got no more biological functionality. Now what I want you to remember is that it doesn't alter the primary structure, right? So when you denature a protein, the amino acid sequence doesn't change, right? The sequence here doesn't change. You've just unfolded it, right? So everything secondary structure and beyond is destroyed. So all those alpha helices would be disrupted, all those beta sheets, everything that maintained quaternary and tertiary structure, that's all trash, right? It's gone. And then it would precipitate out. And then, again, pH is especially nasty because it just destables those IMFs really bad, right? They're charged, amino acids are charged, and so you're gonna have major, major, major problems if you start messing around with pH. And so this is why there's an optimum pH for proteins, right? pH, all proteins are pH specific. So obviously the ones in your stomach aren't gonna have the same functionality as the ones in your cells that are not at pH, 1.5 or whatever pH is in your stomach, right? Each protein is going to have an optimum pH. So the pH, the proteins used in digestion, for instance, aren't going to be the same proteins that you'd find in your eyeball, right? Because they would be denatured at that pH. So I said we'd look at myoglobin and hemoglobin. What's the difference between these two? Do you remember in terms of their structure? Myoglobin has primary, secondary, tertiary. Does it have quaternary? No, it doesn't. Hemoglobin has all four, right? Because hemoglobin has 4 subunits, whereas myoglobin is just 1 subunit. Myoglobin is used for oxygen transport in the muscle. Hemoglobin is used for oxygen transport in the blood. Right, so there's the heme group here in the middle. And so right here is the iron of this shape right here. I'll show you another picture in a second. There's an iron atom here in the middle, and that's where the O2 binds at that active site. So there's looking at it from the top. Okay, so that's where your O2 binds. And so there's that figure I showed you earlier, just looking at it from the side. Right there's your O2 right there in the middle, binding to your heme group. And so when we're thinking about DNA and proteins, again, can we say that all mutations would be bad, all mutations would be good? Well, no, right? It could be a silent mutation, so what would that mean?
It's not effective.
No effect on the protein whatsoever, right? Because if it's, still inserting an arginine where it should be putting an arginine, yes, you're a mutant DNA, but you still got the same amino acid sequence. Let's say you alter the protein and it's not at the active site. Would it matter? Maybe, maybe not, probably not. Let's say it does affect the active site. Could we still say that all are bad? No, because maybe it make the active site better, right? If we go back to our picture of the active site, right? Maybe the substrate now fits better, or this reaction now is faster or more efficient, whatever, right? So even if it affects the active site, you can't assume, if I ask you on a test, hey, are all mutations bad? The answer would be no, right? So you have to be able to explain that, though. You can't just say, no, they're all not all bad, or yes, they are bad, right? You need to be able to explain why. And so this is a genetic modification, all right? You've probably heard of sickle cell anemia. Yes, we're going to do enrichment on Wednesday with looking at DNA modifications and how they affect specific proteins. And so what happens in sickle cell anemia? Do you know anything about that from your biology class?
They have an abnormal curve.
They have that abnormal shape, and what happens?
They don't clot the same way.
They don't clot the same way, but they also don't carry oxygen as effectively, right? And so we're going to talk specifically about this one on Wednesday, so I'm not going to spend a whole lot of time today talking about it. One of the neat things that I thought we would talk about today, because I'm not going to spend time Wednesday talking about it, is, oops, wrong button. I want to talk about, I'm going to introduce it and we'll talk about it more on Wednesday. Hemoglobin transfers oxygen in the blood, right? And so it's totally appropriate for me to be talking about this with a big giant belly, right? When you've got, this is maternal, and there's the fetal, right? In the umbilical cord, where the oxygen's being transferred, O2 needs to go from the maternal blood to the fetal blood, right? Because the fetus has to have oxygen, can't not have oxygen, that would not be good, right? But think about it like this. This magnet over here essentially has to be stronger than this magnet, right? Because if O2 is going to diffuse or go across this membrane, there's got to be something over here that's pulling it, right? Because it's not just going to voluntarily say, oh, I'll just go over here, right? In other words, there needs to be a stronger hemoglobin here than there is here. And so fetal hemoglobin versus maternal hemoglobin. You actually, in your genome, you have two genes for hemoglobin. You have adult hemoglobin, and sometime after birth, you stop making that variant. And when you're a fetus, you make fetal hemoglobin. So in other words, the O2 can transfer from mother to fetus, right? Because this is a stronger magnet. The hemoglobin that the fetus is producing pulls that oxygen over, right? And that's how the fetus gets its oxygen. I mean, because otherwise, without it, the fetus is not going to be able to get oxygen.
Does that make the mother deficient? She's
No.
not affected at all.
No, because I'm still breathing, right? I mean, you know, you're just breathing. I mean, you probably have you do have a higher respiration rate, but this magnet's stronger, so that pulls the oxygen over from mother's blood to fetus's blood without mixing them. And there's a small amount of maternal and fetal mixing, but it's not, you know, they're just dumped in the same, and here's yours, here's mine, right? There's a membrane between them. But that fetal hemoglobin is stronger affinity than the maternal hemoglobin. And so remember when I showed you the lac operon, right? If this is the gene for lactase, right? And then there's a inhibitor that's on the genome when there's no lactose in the cell, right? And then that falls off anytime there's lactose. Well, it'll be the same thing. You've got two genes. I mean, all your cells have the gene for fetal hemoglobin. But once you're not a fetus anymore, you don't make it anymore. And so one of the things we're going to talk about on Wednesday is using this second gene that you have for hemoglobin as potentially a way to treat sickle cell anemia, right? Because if you have sickle cell anemia, if you have all of your hemoglobins are ineffective, right? Because people with full-fledged sickle cell anemia die early in life, right? Because all of their red blood cells look like this. And if we could come up with a way to turn the fetal hemoglobin back on and turn the adult hemoglobin, which is the messed up one, off, then a patient could be treated using their own DNA.
So is that why people freeze cord blood for stuff like this, or is that for something?
That goes to a lot of different kinds of research. Yeah, that goes to all kinds of stuff. But the nice thing, and then we'll talk about this on Wednesday, some of the research is showing that you're using your own blood. You're not taking donor blood. And so that would obviously be better for you because you don't have to worry about the rejection factor. If you could turn your gene for fetal hemoglobin back on and turn your messed up adult one off, right, then you would actually be treating yourself. And so there's the curve, right? This is the amount of oxygen affinity in the fetal version versus the maternal version, right? So if you have sickle cell anemia, it's only affecting the adult version. It doesn't seem to affect the fetal red blood cells. So if you could turn on the gene to have your bone marrow start making fetal hemoglobin instead of adult. And again, we'll look at some research on this on Wednesday. It's really neat. I think it's really neat. Right, so this is what I was saying. It's turned on, it's turned off. But you still have that gene, right? You could be 110 years old. You still have the gene for fetal hemoglobin. It's just turned off. And then we've already talked about this too. Proteins get digested just because you need the amino acids, right? Essential means you can't manufacture it on your own. Non-essential means you can manufacture these on your own. These obviously would have to come from your diet. And I made reference to the movie about dinosaurs. They said they bred them lysine deficient. It's like, well, we're all lysine deficient, right? That's essential for all of us. But, I mean, some of your proteins are just function for diet, right? You break them down so you can use those amino acids in your body. All right, so let's talk about how we classify proteins. Proteins classified by the job that they do. Makes sense. So if you look at a name, or family, I should say, right? All these guys are going to facilitate redox reactions, hence the name oxioreductase. Transferase, move functional groups. Hydrolase, carry out hydrolysis reactions. Lyase, break and form double bonds. Isomerase, we've heard this before, right? Topoisomerase, that chops DNA, but there are other isomerases too, changing the shape. And we've also seen ligase, right? That seals DNA back together. So let's look at examples of each one of these. Here's an oxioreductase, right? So there's the protein that's fused here. And so what you're doing is you're carrying out an oxidation here, right? There's the alcohol being oxidized. Transferase is moving a functional group around, right? So you've got some sort of donor of a methyl group, and now you've stuck it on the end right here. Right? So you've added that functional group, transferase. Makes sense. That's just a figure from another textbook. Here's hydrolase, breaking bonds, right? So look, this will be a triglyceride, right? And the protein here is lipase. And so we're chopping bonds, and so we get our fatty acids and our glycerol back, right? That's hydrolase. And then lyase would be either forming or breaking double bonds. Right, so here we've got a double bond that we are breaking. Makes sense, makes sense. And that's just a figure from another textbook. Isomerases are changing the shape. Right, so you're taking the phosphate group, moving it over here. And ligase, this is the example we talked about when we were doing DNA. Right, this is the example where we're making the phosphodiester bond. This is just for another book. All right, so that's where we're going to stop. Just an introduction to proteins, active sites. On Wednesday, we're going to get into some research on these kinds of things. I really want to talk specifically about the hemoglobin stuff, which is why I don't cover it too much today, because we're going to be doing some activities about it on Wednesday. Do you guys have any question for me? All right, that's where we'll stop.